Welcome to our microlearning channel. Clinical biochemistry is a specialized field within laboratory medicine that focuses on analyzing biological samples to gain insights into human health. This discipline plays a vital role in modern healthcare, from identifying disease markers to monitoring the effectiveness of treatments. By examining substances like proteins, enzymes, and metabolites in the body, clinical biochemists can help diagnose conditions, track disease progression, and assess the impact of therapies. The work of clinical biochemistry laboratory supports physicians in making informed decisions about patient care and management. The field encompasses a wide range of analytical techniques and instrumentation to ensure accurate and reliable results that inform clinical decision-making. Clinical biochemistry is a scientific field that focuses on the analysis and interpretation of chemical components found in the human body. It plays a crucial role in the diagnosis, monitoring, and management of various health conditions. This discipline encompasses the measurement and evaluation of biochemical markers, such as enzymes, hormones, proteins, lipids, and metabolites, to provide insights into an individual's physiological and pathological states. Clinical biochemistry integrates expertise from multiple fields, including chemistry, biology, medicine, and informatics, to deliver comprehensive and accurate laboratory test results for healthcare professionals. Clinical biochemistry plays a crucial role in improving patient outcomes through early detection, diagnosis, and monitoring of various health conditions. By identifying metabolic imbalances, organ dysfunction, and biomarkers, clinical biochemistry enables healthcare professionals to detect diseases early, assess disease progression, and guide targeted interventions for more effective disease management and prevention. Clinical biochemistry supports personalized medicine by providing insights into an individual's unique biochemical profile, allowing for tailored treatment approaches and the monitoring of therapeutic response, ultimately improving patient care and reducing healthcare costs. The information provided by clinical biochemistry testing can help healthcare systems optimize resource allocation, reduce unnecessary procedures, and prevent complications, leading to more cost-effective and efficient healthcare delivery. As clinical biochemists, we are highly trained in the analytical techniques used in the clinical laboratory, such as spectrophotometry, chromatography, and mass spectrometry. Our expertise ensures accurate and reliable test results, which are critical for making the right diagnoses and guiding patient management. By interpreting laboratory test results in the context of a patient's history and clinical presentation, we play a crucial role in the diagnostic process and in guiding patient care. We also conduct research to develop new biomarkers, analytical methods, and technologies that advance the field of laboratory medicine and ultimately improve patient outcomes. Clinical biochemistry involves analyzing various body fluids like blood, urine, and cerebrospinal fluid. These fluids contain many different analytes such as proteins, enzymes, hormones, electrolytes, and small molecules. Measuring and interpreting the levels of these analytes is crucial for diagnosing, monitoring, and managing a wide range of medical conditions. The analysis of these body fluid analytes provides valuable insights into an individual's health status. This slide provides an overview of the importance of body fluid analysis in clinical biochemistry. Accurate test results depend on collecting high-quality specimens. Healthcare professionals must follow standardized protocols to collect blood, urine, or other fluids. This ensures sample integrity and minimizes pre-analytical errors. Collected specimens must be transported to the lab under controlled conditions. Factors like temperature, light, and timely delivery impact analyte stability. Upon arrival, lab staff will process the samples according to procedures. This may involve centrifugation, aliquoting, and storage to maintain quality and prevent contamination. Proper specimen collection is critical to ensure accurate test results. Factors like patient preparation, sample type, and collection technique can significantly impact the integrity of the sample. Appropriate handling of specimens, including transportation, storage, and processing, is essential to maintain sample quality and stability. Improper handling can lead to pre-analytical errors. Factors such as patient diet, medication, exercise, and circadian rhythms can influence test results. Understanding and controlling these pre-analytical variables is crucial for accurate interpretation of test results. Spectrophotometry is a widely used analytical technique in clinical labs. It measures the absorption of light by a sample to quantify the concentration of specific biomolecules like proteins, enzymes, and metabolites. This provides a simple and cost-effective way to analyze a variety of important clinical analytes. Electrochemical methods like potentiometry and amperometry are used to rapidly and precisely measure the activity or concentration of ions. This includes critical parameters like pH, electrolytes, and blood gases. These techniques are essential for applications in critical care settings. Chromatographic techniques like HPLC and GCMS enable the separation, identification, and quantification of a wide range of biomolecules and drugs. They offer high sensitivity and specificity, making them valuable for complex sample analysis. Mass spectrometry provides detailed structural information and accurate quantification of analytes. It's increasingly used in clinical biochemistry for applications like therapeutic drug monitoring and metabolomics. This powerful technique gives deep insights into the molecular composition of clinical samples. Spectrophotometry is an analytical technique that measures how much light a sample absorbs at specific wavelengths. This relies on the principle that molecules and atoms can absorb or emit light energy when they undergo electronic transitions. A spectrophotometer has key components: a light source, a monochromator to select wavelengths, a sample holder, and a detector. The sample's absorbance is measured and compared to a reference, allowing quantitative analysis of analytes in the sample. This technique is widely used in chemistry, biology, and other fields to identify and quantify substances. Electrochemistry is the study of chemical reactions that produce or consume electrons and the associated electrical phenomena. There are three main electrochemical techniques: potentiometry, amperometry, and conductometry. Potentiometry measures the potential difference between two electrodes to determine the concentration of an analyte. This is commonly used for ion-selective electrodes that can detect specific electrolytes. Amperometry measures the current generated when an analyte is oxidized or reduced at a working electrode. This is a sensitive method for detecting low concentrations of electroactive species, useful for glucose, lactate, and other biosensors. Conductometry measures the ability of a solution to conduct electricity, which is directly related to the concentration of ions present. This is often used for measuring electrolyte levels and monitoring processes like water purification. Chromatography is a powerful analytical technique that separates complex mixtures into their individual components. It relies on the differential partitioning of analytes between a stationary phase and a mobile phase. This enables the separation and identification of various substances. Chromatographic techniques include gas chromatography (GC), liquid chromatography (LC), and their subtypes like HPLC and UHPLC. Each type is suited for the analysis of specific classes of analytes. Chromatographic methods are extensively used in clinical biochemistry. They are used for the separation, identification, and quantification of a wide range of analytes in biological samples like blood, urine, and tissues. These include hormones, drugs, metabolites, and other clinically relevant compounds. Mass spectrometry is a powerful analytical technique that allows us to precisely identify and quantify different molecules in a sample. The fundamental principle is that it ionizes chemical species and then sorts the ions based on their mass-to-charge ratio. This technique has many important applications in clinical biochemistry, enabling the analysis of small molecules, proteins, lipids, and other biomolecules. Common mass spectrometry techniques used in this field include liquid chromatography mass spectrometry, LCMS, and gas chromatography mass spectrometry, GCMS. These techniques separate and identify compounds based on their physical and chemical properties, providing highly sensitive and specific results. Enzyme-based assays are a powerful tool in clinical biochemistry, leveraging the catalytic properties of enzymes to measure the concentration of specific analytes in a sample. By monitoring the rate of an enzyme-catalyzed reaction, we can quantify the levels of the target substance. Common enzymes used in these assays include alkaline phosphatase, glucose oxidase, lactate dehydrogenase, and horseradish peroxidase. The selection depends on the analyte and the specific assay design. Many enzyme-based assays use colorimetric detection, where the enzyme catalyzes a reaction that produces a colored product. The intensity of the color is proportional to the analyte concentration and can be measured using a spectrophotometer. This allows for sensitive and accurate quantification of the target analyte in the sample. Clinical biochemistry results rely on high-precision instrumentation that undergoes rigorous quality control checks. Each piece of equipment, from analytical balances to automated pipettes, is carefully calibrated and maintained to ensure accurate and reproducible measurements. Clinical biochemists follow established protocols for quality control and assurance, including regular analysis of control samples, participation in external proficiency testing, and adherence to standardized operating procedures. These practices help identify and mitigate potential sources of error in the lab. Comprehensive quality control programs involve continuous monitoring of analytical performance, including precision, accuracy, and linearity. This data is used to identify trends, detect potential issues, and implement corrective actions to maintain the highest standards of laboratory quality. Reference intervals provide the expected range of values for a given lab test. Based on analysis of a healthy population, these reference ranges can vary based on factors like age, gender, ethnicity, and the analytical methods used. It's important to consider these variables when interpreting an individual's test results. Just because a result falls outside the reference interval doesn't necessarily mean there is a problem. The clinical context and medical history must also be taken into account. The key is to use the reference intervals as a starting point, but not to interpret them in isolation from the full picture of the patient's health. Liver function tests are a key way to assess the health and performance of the liver. We'll cover three main types of tests: bilirubin, liver enzymes, and protein levels. Bilirubin is a waste product from red blood cells. Elevated levels can indicate liver damage or dysfunction. Measuring enzymes like ALT, AST, ALP, and GGT can help detect and monitor inflammation or injury in the liver. Analyzing total protein, albumin, and globulin levels provides insight into the liver's ability to produce essential proteins. These tests give us a comprehensive picture of how well the liver is functioning and can help diagnose and monitor liver-related conditions. Kidney function tests are essential for evaluating the health and performance of the kidneys. These tests measure various biomarkers that indicate how well the kidneys are filtering waste and excess fluids from the bloodstream. Common kidney function tests include measuring levels of creatinine, blood urea nitrogen (BUN), and estimated glomerular filtration rate (eGFR). These provide insights into the kidney's ability to filter and remove waste products from the body. Abnormal results in kidney function tests may indicate underlying kidney disease, damage, or impairment. The clinical biochemist plays a crucial role in interpreting these results and providing guidance to healthcare providers for patient management and treatment. Regular kidney function testing is important for monitoring the progression of kidney disease, evaluating the effectiveness of treatment, and identifying any potential complications or issues early on. Electrolytes like sodium, potassium, and chloride are essential for maintaining fluid balance, nerve function, and muscle contraction. Clinical biochemistry tests can monitor electrolyte levels and identify imbalances that can lead to serious health issues. The body's pH must be tightly regulated to ensure proper cellular function. Clinical tests measure blood gases, bicarbonate, and other markers to assess acid-base status and detect conditions like acidosis or alkalosis. The kidneys play a vital role in maintaining electrolyte and acid-base balance by filtering the blood, reabsorbing essential ions, and excreting waste products. Kidney function tests are crucial for evaluating these regulatory processes. The human body has a complex system for regulating blood glucose levels, involving hormones like insulin and glucagon that facilitate glucose uptake and release by cells. Carbohydrates are broken down through a process called glycolysis, which converts glucose into ADP, the primary energy currency used by cells. Clinical tests like the oral glucose tolerance test can assess an individual's ability to regulate blood glucose levels, which is crucial for diagnosing conditions like diabetes. This slide provides an overview of the key aspects of carbohydrate metabolism, including glucose regulation, glycolysis, and glucose tolerance testing. Cholesterol is a vital lipid molecule that is essential for many bodily functions. It is synthesized primarily in the liver through a complex, multi-step enzymatic process. This process converts simple precursor molecules into the final cholesterol structure. Cholesterol is then transported in the bloodstream to be used by cells throughout the body. Understanding the details of cholesterol synthesis is key to managing conditions like high cholesterol. Proteins are the building blocks of the body, essential for growth, repair, and the production of enzymes, hormones, and other vital components. Protein synthesis is a complex process that involves translating genetic information into functional molecules. The body breaks down proteins into amino acids, which can then be used for energy production, gluconeogenesis, or the synthesis of new proteins. Disruptions in amino acid metabolism can lead to various metabolic disorders, so understanding this process is important. Excess nitrogen from amino acid metabolism is converted to urea, which is then excreted by the kidneys. The liver plays a crucial role in this process. Assessing urea and other nitrogen-containing compounds can provide insights into liver and kidney function. Endocrine function tests are crucial for evaluating the levels of hormones produced by the body's endocrine glands, such as the thyroid, adrenal, and reproductive glands. These tests provide insights into the proper functioning of the endocrine system and can help diagnose conditions like thyroid disorders, hormone imbalances, and metabolic issues. The comprehensive endocrine panel covers a wide range of hormones, giving a holistic view of the endocrine system's health. Thyroid function tests, including TSH, T4, and T3, are commonly used to assess thyroid health and detect conditions like hypothyroidism and hyperthyroidism. Sex hormone assessments, such as tests for estrogen, testosterone, and progesterone, can provide valuable information about reproductive health, fertility, and hormonal imbalances. These sex hormone tests are crucial for diagnosing and managing conditions like polycystic ovary syndrome, menopause, and androgen-related disorders. Therapeutic drug monitoring, TDM, is a critical practice that ensures patients receive the optimal dose of medications. It involves measuring the concentration of drugs in a patient's body to guide dosage adjustments and maximize therapeutic effects while minimizing adverse reactions. Drugs with a narrow therapeutic index, such as antiepileptics, immunosuppressants, and certain antibiotics, are commonly monitored through TDM. This helps clinicians maintain drug levels within the desired range, improving patient outcomes and safety. TDM involves collecting patient samples, often blood or plasma, and analyzing them using advanced analytical techniques like HPLC or mass spectrometry. The results are then interpreted by clinical biochemists to guide dosage adjustments. TDM is particularly valuable in managing treatments for conditions like epilepsy, organ transplantation, and infectious diseases. It helps clinicians optimize drug therapy, minimize toxicity, and improve patient adherence to medication regimens. Toxicology testing is a critical part of clinical biochemistry, used to detect the presence and concentration of drugs, medications, and toxic substances in biological samples. These tests help identify and monitor potential poisonings, overdoses, and substance abuse. Drug screening panels test for the presence of illicit drugs, prescription medications, and other substances. These tests are widely used in healthcare, law enforcement, sports, and workplace settings. Common analytical techniques include immunoassays, chromatography, and mass spectrometry. These methods offer high sensitivity, specificity, and the ability to quantify analyte levels. Interpreting toxicology and drug screening results requires expertise in clinical biochemistry, pharmacology, and toxicology. Clinical biochemists work closely with healthcare providers to ensure accurate interpretation and reporting of results. Pediatric and geriatric patients have unique physiological differences compared to the general adult population. These differences affect how drugs are metabolized, fluid balance, and normal reference ranges for various biomarkers. Specialized testing and interpretation is required to account for these age-specific variations. Clinical biochemistry tests must use age-appropriate reference intervals to accurately interpret results for pediatric and geriatric patients. Factors like growth, development, and aging can significantly impact normal values for analytes like hormones, enzymes, and electrolytes. Obtaining adequate specimens can be more difficult in pediatric and geriatric patients due to small blood volumes, fragile veins, and cognitive/physical impairments. Careful technique and specialized equipment are essential to ensure reliable test results in these patient populations. Point-of-care testing, POCT, allows for rapid analysis of patient samples at the bedside or in a clinic, providing immediate results without the need to send samples to a central laboratory. POCT enables faster diagnosis and treatment decisions, leading to improved patient outcomes, especially in critical care or emergency situations where time is of the essence. POCT devices can be deployed in a wide range of settings, including primary care clinics, pharmacies, and even patient homes, expanding access to diagnostic testing. The ease and convenience of POCT can enhance patient compliance and engagement, as they no longer need to travel to a lab or wait for results. Metabolomics is a powerful technique that can uncover novel disease signatures and identify early-stage indicators of health conditions. By analyzing the comprehensive profile of small metabolite molecules in biological samples, metabolomics research is leading to the discovery of emerging biomarkers. Exosomes, tiny extracellular vesicles released by cells, are proving to be valuable biomarkers. Analyzing the cargo within exosomes can provide insights into disease processes and enable the development of non-invasive diagnostic tools. Epigenetic changes, such as DNA methylation patterns, are emerging as powerful biomarkers that can offer insights into disease predisposition, progression, and treatment response, opening up new opportunities for precision medicine. Advances in genomic technologies allow us to analyze a person's genetic makeup in great detail, identifying genetic variants linked to disease risk, drug response, and disease progression. Innovative biomarker research is uncovering new molecular indicators that can help predict, detect, and monitor disease, enabling more personalized treatment approaches. Precision diagnostics allow us to select targeted therapies tailored to an individual's unique genetic and molecular profile, improving treatment outcomes. This personalized approach to medicine holds great promise for delivering more effective and efficient healthcare. The future of clinical biochemistry is being shaped by rapid advancements in automation and digitization. Laboratories are embracing robotics, AI-driven data analysis, and cloud-based information systems to streamline workflows and reduce errors. These technological innovations are transforming the diagnostic landscape, making clinical biochemistry more efficient and accurate. As our understanding of genetics and molecular biology deepens, clinical biochemistry will play a crucial role in personalized medicine. Tailored diagnostic panels and precision-guided treatments will become more commonplace, allowing clinicians to deliver targeted, patient-centric care. Portable, user-friendly point-of-care testing devices will continue to proliferate. These rapid diagnostic tools will bridge the gap between the laboratory and the bedside, empowering patients and healthcare providers to make more timely, informed decisions. Ongoing research is uncovering novel biomarkers, expanding the diagnostic arsenal of clinical biochemistry. Innovative techniques like metabolomics, proteomics, and liquid biopsy will enable the identification of disease-specific signatures, leading to earlier detection and better-informed treatment strategies. Clinical biochemistry professionals must adhere to strict ethical principles, such as respect for patient autonomy, beneficence, non-maleficence, and justice. Maintaining confidentiality and obtaining informed consent are critical to upholding these ethical principles. The field of clinical biochemistry is subject to various regulatory bodies and guidelines, such as the International Federation of Clinical Chemistry and Laboratory Medicine, IFCC, and national accreditation agencies. Adherence to these regulations ensures standardized practices and reliable test results. Clinical biochemists must stay up to date with the latest ethical and regulatory developments, participating in ongoing training and professional development. This commitment to continuous improvement helps maintain the highest standards of practice and patient care. Clinical biochemistry is a vital field that provides essential insights into a patient's health by analyzing various body fluids and metabolic markers. It plays a crucial role in disease diagnosis, treatment monitoring, and personalized care. As technology advances, the field of clinical biochemistry is poised to evolve, with the emergence of new biomarkers, point-of-care testing, and personalized diagnostics. These advancements will enhance the speed, accuracy, and individualization of clinical decision-making. Clinical biochemists must navigate complex ethical and regulatory frameworks to ensure the responsible and ethical use of diagnostic tests, safeguarding patient privacy and promoting the highest standards of care.